Assalamu alaikum. So today we're in this class we're going to talk about the histological features of cerebellar cortex, and this is basically histology practical. But let me just give you an overview of what cerebellar histology is, and then I'll just show you the slides which are present in histology lab at Ziauddin University. So in case अगर ये paper में आ जाती हैं, तो you'll be able to identify them. So we'll just extract some identification points if they ask you to write, and I'll also tell you about what they can ask you in exam. So first we'll talk about the overview of the gross features, as you all know, and then we'll talk about the histological features of the cerebellum. And in histology, we'll talk about the cellular components as well as the layers of the cerebellar cortex. So when we talk about the cerebellar cortex, it's basically divided into cellular component and the layers. First, let's talk about the cellular component. So there are four types of cells that you can find in cerebellum. One is known as the granular cell, Purkinje cell, basket cell, as well as Golgi type 2 cells. Out of these four cells, these two cells are the major components of the cellular aspect of cerebellum. That's why the layers that we write when I'll talk about that. सो ग्रैनुलर लेयर एक पूरी बनी हुई होती है, इसमें मेजरली ग्रैनुलर सेल्स पड़े हुए होते हैं. एक लेयर होती है जो स्पेसिफिकली हमने परकंजी सेल सबसे ज़्यादा अबंडेंट होते हैं उस लेयर में, सो उसको हम परकंजी सेल लेयर बोलते हैं. As well as compared to basket cells and Golgi type 2 cells, these are the cells which can they're scattered all over. They're usually present within all these layers, and ये बिल्कुल minor cells को consider कर सकते हैं. तो majorly जो functionality की बात करते हैं about cerebellum, and cerebellum works as turn on and turn off mechanism. So majorly Purkinje cells and granular cells come out. As far as the functionality is concerned, all of these cells are inhibitory except the granular cell. Granular cell is the only cell which is excitatory in nature. So whenever this cell is going to be excited or stimulated, it's going to generate an excitatory impulse. While Purkinje cell, basket cell, and Golgi type 2 cells, they all are inhibitory in nature. So let's talk about granular cell first. Granular cell, as we all know, the name suggests that they should be globular in shape. They're present majorly in the granular cell layer, which I'll just tell you about later. They're darkly stained, as you can see. Because why they're darkly stained? Because they have more nucleus and very less amount of cytoplasm. So this is all nucleus that you can see, and it's a very less rim of cytoplasm you can see here. So that's why they're darkly stained, and they have multiple dendrites that you can clearly see here. This is one dendrite, this is another dendrite, this is other. So they have multiple dendrites, and the fiber that goes and excite these cells are basically the fibers that coming in are basically mostly mossy fibers, which majorly when physiology people will tell you about the functionality of cerebellum. I don't know, they already have told you about it. So if you know about it, there are two types of fiber, climbing fibers and mossy fibers. Mossy fibers are basically responsible to excite the granular cells. Then we have Purkinje cells. Purkinje cells are basically flask-shaped cells. You can clearly see here. So this is Purkinje cell, and they're majorly present in Purkinje cell layer. And the fiber which comes and excites or stimulate this cell are basically the climbing fibers, and they're inhibited by the basket cells. When we talk about the basket cells, you can clearly see here, this is the Purkinje cell, and you can clearly see in very close proximity, you can see another cell that's called the basket cell. Basket cell can be present in molecular layer as well as Purkinje cell layer, and it surrounds the Purkinje cells like a basket. That's why we call them basket cells. And they send inhibitory signals to inhibit the Purkinje cells. So whenever a climbing fiber comes and excites the Purkinje cell, they can come and inhibit it. So that's how it works. Then we have Golgi type 2 cells, which are again they're majorly present in the granular cell, but they can also be present somewhere around in the molecular layer as well. And they have more cytoplasm. And again, they synapse with both mossy fiber as well as dendrites of the granular cell. So they inhibit the granular cell exactly like the basket cells were inhibiting the Purkinje cells. So this was all about the cellular component of the cerebellum. So now we talk about the layers. This is where we can extract some points of identification that you need to identify the microscopic slide of cerebellum. So this is a diagram that I've taken from Diaforce, and this is schematic diagram, so it's a drawn diagram. So you can clearly see the three layers. So the outer I'm coming from outside and I'm coming inside the organ. So in the cerebellar cortex, the first layer that you can see is the molecular layer. So the molecular layer, you can clearly see that there are a few basket cells that you can see here, and all you see is the fibers of the Purkinje cells as well as the granular cells. And then comes the Purkinje cell layer, which is a single row of Purkinje cells. So they're not multiple; they're just standing in one line. It's a single layer, so it's very thin. And then when we talk about the deepest layer, that's called the granular cell layer. It's known as, you can clearly see it's darkly stained, obviously, because the granular cells itself are darkly stained and they're more like they're abundant. So that's why it gives a very dark stained area. So from outside to inside, the first layer is basically the molecular layer, second layer is the Purkinje cell layer, and the third layer is the granular cell layer. And once we go further inside, then you can find the cerebellar medulla where your deep cerebellar nuclei are present. No, so this is basically the H&E stain. Overall, you can clearly see the three layers. You can clearly see the molecular layer. I'm mostly focusing on the molecular layer in this slide. So it's the outermost layer, as I just told you, contains very few cells, mostly basket cells are present there. It has no myelination. There are no myelinated fibers there. Contain the parallel fibers that are coming from both granular as well as Purkinje cells. Purkinje cell layer is just a single layer, it's right in the middle. It's a single layer, it's a single row of the cells, as I told you earlier, and like stellate shaped Purkinje cells, which you can clearly see. This is one Purkinje cell, this is another Purkinje cell, this is another Purkinje cell. So, and then when we talk about the granular cell layer, it's the innermost layer, as I told you earlier, and since it appears as closely packed region, therefore it gives a very darkly stained area, because obviously the granular cells are also darkly stained because of the increase or a larger nucleus compared to the content or the amount in the cytoplasm. So it contains the nuclei of the granular cells, and it contains the empty spaces in between the those cells. They are known as the glomeruli of the island. As you can see, the white spaces here. These are the glomeruli of the island, and then these islands, they contain the dendrites of the granular cells that receiving fibers from the outside. So this is the slide that I took picture of from Ziauddin University histology lab. So you can clearly see here, this is the molecular layer, that's the outermost layer, which you can clearly very less number of cellular component. Then you can clearly see a row of Purkinje cell right here. You can clearly see them. These are the Purkinje cells, and then this entire dark area or the darkly stained area, this is the granular layer of the cerebellar cortex. So as far as the points of identification is concerned, you can comment on the layers, you can tell about the cellular components, you can tell about the staining, which area is stained darker compared to the other area. You can talk about the single row of the Purkinje cell. So there are so many points of identification. We'll only ask you about like 2 or 3 points of identification of cerebellar cortex. So this is the slide. If you put, so you can easily identify. If you see three layers, that's definitely cerebellum. Furthermore, that's another slide I had. This is again pretty much clear. You can clearly see the molecular layer right here. You can see these Purkinje cells, and this entire thing is basically the granular cell layer. And then this is a specialized stain. एक picture में ली थी, clearly demarcated. That's a super magnified picture of stain. This is a silver stain, by the way. So this is the molecular layer. You can see the fibers here. This is a specialized type of stain that we do to enhance the fibers as well. Because in the previous slide, you can clearly see the cell bodies, the nuclei, but you cannot see the fibers. But in this, you can clearly see the fiber. These are the fibers of the Purkinje cells which are coming into the molecular layer. This is the molecular layer. This would be the basket cells, and these are the Purkinje cells right here, flask shape, and this entire thing is basically the granular layer. So these are the granular cells. Maybe I don't think I can see a basket cell here, but it'll probably definitely be here somewhere around. Maybe this is a basket cell that's inhibiting the fiber of the Purkinje cell. So this is all about the cerebellar cortex, and if you have any question, you can ask me through any means, and this is it. Thank you.